So yeah, I will present a few projects I did during my PhD, also some of my colleagues' work, all around using AI for peptides that are somehow involved in the immune system. So for those without a background in biology or anything related, it all starts when a pathogen, so for example a virus or a bacteria, enters the human body, or when some cancer develops. Then in the body there are systems to cut these pathogens or proteins from the cancer cells into small parts, and these parts are called epitopes. So these are very short pieces of proteins from the pathogens. So that's what we have here. Then in our immune system we have T cells, and these T cells have a T cell receptor, and that is a very important part because almost all of the T cells in our body have a different T cell receptor, and these different T cell receptors can then recognize or bind to different epitopes. So for example, that T cell would recognize this pink epitope, but the other T cells don't recognize it. If you look a bit in more detail at the interaction that happens when they bind, so on the left is just a small part of the T cell receptor from the T cell, and we have one important region, the CDR3 region, that we know that is always in very close contact with the epitope. It also has a lot of variability, so the other parts of the T cell are the same for almost all T cells, but the CDR3 region, that is actually the thing that is different in almost all T cells. And then on the right is the epitope, so the short piece of protein from the pathogen or cancer cell. So because both of these are protein or peptide sequences, we can also represent them by their amino acid sequence, and each amino acid can be represented by a single letter. So to make it a bit more easy for us computer scientists to work with it, we just represent the CDR3s as a sequence of letters and the epitope as a sequence of letters. So when we go back to here, the moment this epitope actually recognizes this T cell, the other way around, this T cell recognizes this epitope, the T cell will get activated and expand. So it will make many copies with different functions that will eventually kill the pathogen and will lead to the immunological memory, so that you are protected from that pathogen in the future. So our goal is to have a machine learning model that can predict whether a given combination of T cell and epitope will lead to this activation or not. This is beneficial for healthcare. For example, we can use it to develop new vaccines or to make cancer treatments. So the first part I want to talk about is some explainable AI we did on these prediction models. So we used two of these prediction models, Immrex and Titan, that make these predictions, as I explained, and we want to extract which features from the input sequences they used to make these predictions. So to put it a bit simpler, we want to know for a single example which letters were important for the prediction and which letters were not important. What we then did with these predictions is we show them on the three-dimensional structure. So here the blue is a high feature importance, so it means was important for the prediction, and green is not important. So on the CDR3 it's mainly the amino acids in the middle that were most important, while for the epitope here it were mainly the amino acids on the bottom or the start of the epitope. On the other hand, the Titan model, so this is a completely different model, but trained on the same data, learned something completely different for this example. So they did not really look at the CDR3 region, they only looked at a few positions in the epitope. So when we then do this for a lot of examples and take the average attributions per position. So here, these E's are the positions of the epitope, the C's are the positions of the CDR3. Then for the epitope, IMREX looks a bit more at the beginning, Titan a bit more at the end. But what is much more surprising is that Titan is not looking at the CDR3 region, but only at the epitope, while IMREX looks at both of them. So this is a bit weird, because we want to solve an interaction prediction problem where we assume that both parts, of course, are important to determine the interaction. And if you then quickly look at the performance, you see the performance is not good, but it is still similar for both. So how is this possible? We found two problems that contributed to this. And for this, I will first explain you how the data is constructed. So we don't have negative data for this problem. We can only get positive samples, so T-cell epitope combinations we know that bind. So what we usually do is to get the negative data, the open circles here is we will randomly shuffle T-cell epitope combinations to get random T-cell epitope combinations, and because there are so many different possibilities, we assume that the random combination will always be a negative example. So that is what we did both in train and test data. The different colors are different samples for the same epitope or for the different epitopes. Then in Titan, they do this data generation or negative data sampling a bit different. So instead of keeping the same number of positive and negative samples for each epitope, they just took the same number of negative samples for each epitope. I said the same thing twice, but yeah, I think you can see what is happening here. And this actually introduces a bias. So for some epitopes, there are more positive than negative samples in the trained data set, while for others there are more negative than positives. And this is a problem because the machine learning model will learn this bias. There was also a second problem. And this problem was the train test strategy. So in green is what IMREX does. It is a machine learning model that is trained for 20 epochs, and then the performance or the model of the last epoch is used to get the test performance, while Titan would make test predictions each epoch, and then at the end just report the performance of the best epoch, which is of course not a correct way to evaluate a machine learning model. So eventually what we think happened in Titan is that they would each epoch select some random pattern in the epitope, which is also what we saw in the feature attributions. And then when this pattern by chance corresponds to a pattern in the test data set, this will give a very high performance, and then because of the second problem, that performance is reported. In the second part, so this is actually similar research, but on a different model. So here we saw last year that a new model was published in Nature Machine Intelligence that also tried to solve this problem, and suddenly they reported a performance of almost 71% compared to previous models that were all around 55% only. So as you see, it's a big jump in performance, and we wanted to investigate how this was possible and whether this was actually true. Unfortunately, we again found a problem in the negative data sampling. So this time the problem was a bit different. In a bit more detail, what we usually do is this. So we shuffle the T cells, the squares, and the epitopes, the circles, from the positive data set to get the negative data set. What PAMPEP here, this new model, did instead was pair the epitopes from the positive data with completely different TCRs from a background data set. And the problem here is that this background data set, these TCRs, this data, is often generated in different labs from completely different type of samples and have a slightly different background distribution, which leads to something called shortcut learning, where the model, instead of learning the true things that determine which pairs bind and don't bind, will just learn the difference between the positive and the negative TCRs only. And when we then would eventually apply it on different type of data where the negatives are not made in this way, then yeah, suddenly the performance would be much lower. So that is what we tested then. So we used the PAMPEP model, test again with their positive data, but the negatives generated with the shuffling method instead of their method. In theory, if their model would work good, it should be able to make good predictions on any correct way that the negatives are generated, but we saw that for them this was not true. Suddenly the performance is completely random. So this is also not that surprising, because if you introduce a bias in the data, this is the simplest way for the machine learning model to distinguish between the positive and the negative samples. So yeah, they will always go for the simplest solution and just learn the bias instead of the true patterns. Okay, after the second part, I think we can already do some questions, because the following parts are a bit different in topic. So if anyone has any questions about any of the first two parts, we can have them now. Yes?
I had a question about the last slide you showed about the performance dropping. Did you retrain their new model? Did you just...
No.
If you have to understand it.
No, so we used their pre-trained their model, tested again on their data set, but instead of generating the negatives with their method, we generate the negatives with the shuffling method instead. And yeah, as I said, if the model actually learned the difference between positive and negative samples, it should still work on our data, or on the data where the negatives are generated with our method. But this was not the case. They just only learned the bias in the data set that was caused by how they generated the negatives. Are there any more questions? Yes.
Yeah, about the comparison between Titan and the other technique. There was a slide about the early stopping of Titan over here. So what's your take on, you said it's not done to do an early stopping and to take the the epoch with the highest accuracy? Is it because it's there's more instability, so the method is less robust, or?
Yeah, so you could do it if you had an additional validation split, and then you would stop when the validation performance, or take the epoch of the best validation performance. But yeah, in this case, as you see here, and this is mainly because of the first problem, that the performance is just random, going up and down, and then yeah, performing the test performance of the best epoch is just random, where yeah, we think that then just by chance some pattern Titan found in a trained data set that corresponded between this imbalance between positive and negative samples could also be applied to the test data set, and then by chance had a very high performance. But if you would apply it on a different data set, it would not have performance anymore. Okay. Good. There are some more questions.
Yeah, maybe I missed it, but how do you make a truly randomized sample, the negative bias, without bias?
So maybe I will have the more detailed. So we just shuffle the epitopes from the positive in a different order, making sure that we don't create pairs that were already positive, and that is for our purpose enough.
So how do you avoid to unintentionally have structure in the negative data that maybe the model can learn? Because if you randomize, for example, with pseudo-random numbers, maybe the model can understand the pseudo-randomness rather than random.
I think that's not really possible. So at the moment we're talking about 20 to 40,000 samples we have. The model is not really able to even learn any true patterns that are in the model, or not that well, at least, as you saw here on the performance graph. So I don't think the model would be able to learn anything about the random generator. If there are no more questions, I assume. Okay. In the next part, I will be talking about the techniques to actually get the epitopes that are presented by or to the immune system. So here we have a cancer, and what happens, the cancer will, or the cancer cells, all cells, not only cancer cells, will present the epitopes I've been talking about before on their cell surface, that can then be recognized by T cells to attack the cancer. But as you know, often in cancer, this is not enough. Our immune system cannot kill the cancer quickly enough, and we can help the immune system by identifying which of these neoepitopes, they are called then, are represented to or presented to the immune system, and in this way we can give the immune system a boost to actually kill the cancer. So we identify these epitopes with mass spectrometry. In the field of mass spectrometry, we usually call them peptides. And so from a sample, this can be a blood sample, tissue sample, or other fluid sample, we want to know the sequences of the peptides that were in the sample. But this machine does not immediately output the peptide sequences, it outputs these spectra. And then our goal is to identify the the peptides from the samples based on the spectra, and this is traditionally done with database search. So here we have a database of all sequences that we expect might have been in the sample, and from those we compute the theoretical spectrum. And then we will compare the experimental spectrum with all the theoretical spectra, and the one with the highest similarity will then be the match, and will lead to the peptide sequence that was in the sample. But unfortunately, this method only works for a limited number of peptides, and for most of them, the data is not good enough to actually identify the peptide. So what we do in our lab is we use machine learning techniques to predict properties of peptide sequences. So for these models, the peptide sequence is the input, and then some properties that are also related to the mass spectrometry that are usually outputted also by these machines are predicted to then combine with the results of the database search to get an improved score and a higher number of peptide identifications. Okay, in the first project, we have been looking at these peptide prediction models and the influence of the size of the training data. So here this was a transformer encoder model, and as you know, often more data is better. So this was also here the case. When we go to larger data set size, the error decreased, and we even expect that more data can still improve our models. Then a second thing we tried was pre-training these transformer models. So this is done by masking part of the input tokens, in this case amino acids, and then predicting them, or learning the model to predict them. And we found that this also helps the predictions. So again, the error, so lower is better on the left, the model that was not pre-trained; in the middle, a model that we just pre-trained on the same data we have for the fine-tuning, so the actual task. And then on the right, we use a model that was pre-trained on a very large general protein data set. And we see that even doing the pre-training ourselves helps, but using a large protein data set for the pre-training does slightly better. So these peptide property prediction models often work very good in simple cases, but there is a problem. Proteins very often, or almost always, have modifications, and these are small, sometimes large changes on the amino acids that make that the proteins have different function and will also influence the peptide properties. But the machine learning models we trained on these peptide sequences do not work anymore with these modifications. So the solution we came up with is, instead of just representing the peptide as a sequence of tokens, representing them by their molecular structure. And in this way, we have a few advantages. So we can encode any modification. It doesn't have to be predefined. As long as we know the change in molecular structure, it's enough. And this model also has very rich input information about the amino acids and their structure, and can hopefully learn how this influences the properties we want to predict. And then the sequence of molecules can be, for example, embedded with a molecule embedder, and then a transformer model can learn the peptide property prediction. because we saw that this is rather difficult and a lot of information for a single transformer model to process and learn, we thought of doing a two-step approach where we first start with predictions for the unmodified peptide, because we know that they are very good and very accurate. And then this prediction is added as an additional input for the model I showed in the previous slide that will then predict the shift, so the difference between the prediction of the first model and the actual peptide property. If we then look at the results, so on the left, the errors over all test samples, and here, to make it a bit more difficult, we test on peptides that have some type of modification that was never seen before during training. So, wrong button. So this means that the input will have molecules that were not seen during training. So it's rather difficult, but in green is a model that previously existed. In purple, the one step, the default molecular structure transformer, and then in orange, the two-step model, and we see that the two-step model can actually do quite a lot better. On the right is just the performance, but first grouped per PTM. The PTMs are modifications. Okay, then in a slightly different project, I want to focus more on the spectra itself. So some of you might know or might have noticed that these theoretical spectra, they all have the same intensity. So it is not really possible to compute the intensity of these spectra theoretically. So to improve the identifications even more, we want to predict them, because as an example, we have an observed spectrum, so both is the same spectrum we observed on the top, and on the bottom we have two different theoretical spectra, but from this we have no idea which one is actually the correct one. So if we then add the predicted intensities, we clearly see that the right matches much better than the left. If we want to do this for immunopeptidomics, so to identify the peptides from the immune system, it is a bit more difficult, because we often have a very low abundance of these peptides in samples, and there are different types of mass spectrometry. The one in the top, if you look all the way at the right, has some difficulty with identifying or measuring these high mass peaks, while the different method on the bottom can still measure these even for single cell data, so very low abundance data. So if you want to look at these epitopes of immunopeptides, we need the thin stuff method to get the data. This is also what was shown here. So in the dark color is the previous more simple method, and the number of peptides they were able to identify. In blue is the peptides that both methods were able to identify, and then the timsTOF we are looking at in green, and we see that they add a lot more peptides to be identified. But I've been talking about this intensity prediction. If you look at a spectra from the same peptide measured on both machines, it's very different. So if we do these intensity predictions, it's logic that it will need to be different for both techniques. So we used a previous intensity prediction model and fine-tuned it on timsTOF data, because there was no model available yet for this timsTOF model machine. And we see that this made the spectral angle, so this is like a similarity metric for spectra, much higher on this type of data. We actually use these predictions to rescore, so MaxQuant is a database search method, and then we incorporate these intensity predictions, also the peptide, so all the peptide property predictions I've been talking about in the rescoring, and there we see that this now adds a lot of peptides that can be identified from different types of samples. Comparing the Orbitrap with the timsTOF method, again, we see that using rescoring on timsTOF gained a lot more peptides than on Orbitrap, which is a good thing. And then on a different data set where we compare the number of cells with the number of peptides that can be identified, and again using rescoring with our fine-tuned model increases the number of peptide identifications. And then to check that the peptides we add are actually correct, we can look at whether they would actually be presented to the immune system. So we know that all peptides from a certain HLA molecule will have this motif on the left. And if you look at the one that were found by both methods, this motif corresponds very well, so that is as expected. The ones we gained with our method also have the same motif as the motif we expect, so that is also good. And then the ones we lost, so that our method did not find, and the previous method did find, we see that they don't correspond to the expected motif at all. So this is also a good thing that the ones we lost are actually not good peptides or peptides we want to find. Then this is something similar, so we look at the chance that these peptides will be presented to the immune system. So more to the left is better, and we see that the ones that are shared and gained are very high proportion has a lower affinity score, while the ones we lose are mainly high affinity scores. Okay, I have some time. Then in the last part, we again want to identify peptides from spectra, but instead of going through all the database search and rescoring, we want to do it directly. So what we do is we use the transformer architecture to actually translate the spectrum immediately to the peptide sequence. So in the encoder, we input the spectrum by using tuples of mass and intensity of all the peaks, and then in the decoder we also add the total mass and charge of the measured peptides, and then as usually done with these transformer decoders, we will predict the next token one by one given the previous sequence. And then at the end, there is a beam search algorithm that also takes into account all the previous predictions of all the tokens. Okay. So here, DeepNovo and Novor are existing models. Casanovo is the new transformer-based model. The one in blue trained on 1 million sequences. The one in the dark pink trained on 30 million sequences. And we see that we can do a lot better than the previously developed methods, and also that a large data set size really makes a difference. A problem we often have is that there is a lot of triptych data, it's called. That's a certain way of generating this data, but this cannot or can often not be used for the immune system peptides or the epitopes, because this type of data looks a bit different, and this is also what we see here. If we use Casanovo on the immune data, it performs much worse. So we fine-tuned the model on non-triptych data, on immune system data, and this already does better, but it's still not as good. Then again, similar as before, we did some evaluation to actually check that the predictions make sense. Here, Tide is a database search method, and we see that the ones that are shared between Casanovo and Tide and the ones that are only predicted by Casanovo have a very or have a lower binding affinity, which means that they are probably true, while the one predicted by Casanovo only by Tide have a higher binding affinity. Okay, then I just want to thank my lab in Antwerp, some people from Canada and Munich that provided the data, and also people from Washington or the University of Washington in Seattle that worked together on some of the projects.